All right, people are still joining. I guess we will start the class. So, good afternoon. We will continue our discussions on cloning, right? Let me share my slide. So, are you able to see the slides?
Yes, sir.
Okay. Okay. So, when we look at cloning very broadly, we can have three different types of cloning. One is molecular cloning or gene cloning, which creates copies of genes or segments of DNA. Okay, it could be any segment of DNA. Okay. Reproductive cloning, which creates copies of whole animals or plants. Okay. And then you have therapeutic cloning, which creates embryonic stem cells, and these are essentially somatic stem cells that are reprogrammed to cure people with Parkinson's disease or Alzheimer's disease. Okay, because what happens in these patients is that they have specific pathology associated with the brain, and you can reprogram these your somatic cells, right? You can take it from the patient's own cells. You can reprogram it, and then from a skin cell it will become a neuronal cell, and then you can implant it back into the brain of these patients, and it is gives a lot of therapeutic benefits. So that is called as therapeutic cloning. Okay. We have seen gene cloning, right? We have spent considerable amount of time the last, maybe, I don't know, since we started. I guess I think we are today in 13th class, or this thing. So last 10 classes or so, we looked at how genes can be cloned, how it can be expressed, different expression platforms and all that. Uh, what are clones? Clones are nothing but identical molecules. It could be organism or it could be molecule which is derived from a common ancestor or from a starting material. You're going to make copies of it. You're essentially making copies of it. It's like something that we are all familiar with. We have, say, a typewritten page, then you go and photocopy it. Photocopy maybe 100 copies, and so then you won't see any changes in it, right? Photocopies, you're not going to see any changes in it. It is something like that. Okay. So when it comes to copying DNA molecules, it's called as molecular cloning or gene cloning. When it comes to plants, either you can generate whole plants by various methods, asexual reproduction, tissue culture, and then putting them into specialized media, you can generate the whole plants. When it comes to animals, two very commonly used methods are SCNT or single cell nuclear transfer. Then you have embryo splitting, a developing embryo at a very early stage, very early, not at a late stage, but at a very early stage when they are still in the form of two cells or four cells, you could split them. Generally, embryo splitting is done at a even earlier stages. When it is just a single cell zygote, people, you can split that into two, and you're going to get identical twins. Okay. And then you can have stem cells, which is nothing but taking it from your own somatic cells. You can reprogram them. All your cells, by and large, except your hepatocytes, which are in the liver. Most of these cells are terminally differentiated. That means there is no more, they can't go back to the embryonic state, but with special cocktail of transcription factors and special growth media, these terminally differentiated cells can come back to an embryonic stem cell like condition. Okay, that's why these are called as embryonic stem cells. And when you do it with your somatic cells, then it is called as induced pluripotent stem cells. Okay, so we are not going to deal too much about stem cells, except saying that there are two kinds of stem cells. One is called embryonic stem cells, which is derived from fetal embryos, and one which is called as induced pluripotent stem cells, which are derived from adult cells which are reprogrammed to acquire embryonic stem cell like characteristics. Okay, any doubts on the stem cell part? Can we go ahead?
Yes, sir.
Okay. All right. So cloning plants from single cell, we all know that, for example, two of the most commonly or very popular these things is carrots and tobacco plants, okay, which can be very easily cultured in the plant tissue culture medium. So in this case, for example, from a carrot, a plug of tissue is removed. These are disassociated into single cells. And these single cells themselves are transferred to different growth medium where they divide to form callus, right? So callus is something you don't call this as callus in animal cell culture. So I think the terminologies are little different between plant culture and animal culture. Okay, so these grow into callus, and each callus in turn, with appropriate medium, can be made to grow into a whole new plant. Okay, so this is very very useful when we are talking about some very rare, say for example, some rare herbs or some plants which are about to be, which are endangered and which need to be rescued, right? So plant tissue culture comes into this thing play in those situations. Okay. Since you have a whole lot of this thing on plant tissue culture and those applications, I'm not dwelling too much into the plant. I'm going to focus on animals. Okay. So animal cloning has been going on, for a fairly long time. It's I think although it says 20 years, I guess it has been more than 20 years, since people started practicing this animal cloning. Why do you do animal cloning in the first place? When we see some characteristic which could be of benefit, then people start cloning. For example, the Australian sheep, the wool from Australian sheep is supposed to be the best quality. So people wanted to clone. It didn't happen all of a sudden. I think it happened through selective breeding, and then people started cloning those things. So there are two things. One is called embryo splitting, which as I said, and then you have another one called nuclear transfer. So typically, what happens is the embryos split to form two half embryos, and then embryos are transferred to a surrogate mother, okay, who is not related to this thing, okay, and then the pregnancy is monitored by ultrasound, and then you have the surrogate mother giving birth to the babies, right? um, so they give rise to identical. In this case, because the embryo split into two, you got identical twins. Okay. So the eggs are collected, they are fertilized. In reality, how embryo splitting happens is that the eggs are collected, they are fertilized by in vitro fertilization. That means it happens under laboratory conditions. Okay. The embryo is grown to 8 to 16 cells, and then the cells are separated. Okay, and each of those cells that are separated now, depending on which stage you're separating, whether it's a 8 cell or 16 cell, each of these cell have now the potential to grow into a whole embryo. That means from one fertilized egg, you now, instead of having one zygote, you can now have at least 68 to 16 different progenies coming out. Okay, so these embryos are then transplanted into surrogate mothers, and this ideally can be used to clone any mammalian embryos, including human. Okay, so that means the procedures have been fine-tuned to such an extent that even this process is applicable to humans. I mean, in vitro fertilization was once upon a time a very very rare and advanced, very very sophisticated and advanced technique, right? But today you can see literally, every, the in vitro fertilization for couples who are not able to bear children by normal means has become so routine that, every state Every district has one IVF center or maybe multiple IVF centers, in vitro fertilization center. It has literally mushroomed. IVF has mushroomed across India like anything, right? So we know that somehow these things are part of this thing. Okay, although IVF by per se is not this thing. The IVF that is done, in vitro fertilization that is done, is different. But what I'm saying is the procedure has become so common that it could be used for cloning. Okay, so that was about embryo splitting, right? We don't do that with humans. We don't do that. Let me reiterate that we don't do that with humans. In fact, even with animals, it is done very rarely. Nevertheless, that is a possibility. Okay. So how does cloning by nuclear transfer happen? This is how Dolly the sheep was born. Okay. So these cells, there is a donor and there is a animal which supplies the unfertilized egg. Okay, the egg turns out to be the crucial component. Okay, so from the egg you take out the nucleus. Okay, you take out the nucleus. This nucleus, as we all know, is haploid, right? Haploid, and when it meets the sperm, it becomes a diploid. So it becomes 2n, right? Now, after you remove this nucleus, okay, you take any other organism which you want to clone, okay, and you take a somatic cell which has 2n, right? The nucleus has 2n content of DNA, okay. You take the nucleus, okay, and then you inject this nucleus into this egg, okay, from which the nucleus has been removed, okay. This incoming nucleus has 2n number of chromosomes, right? It is restored, okay. For some reason, the moment this 2n number of this thing gets into the nucleus, the egg behaves as though it got fertilized, and that's how it starts dividing, okay. And it starts dividing, and you have, because the nucleus was derived from this donor, you're essentially creating a clone of this donor, okay. And then you have a cloned animal which is exactly, exact DNA as this donor, okay. So this mother, the nucleus serves as a surrogate, okay. This itself is after transfer of this nucleus, into the egg, this itself is implanted into a surrogate mother, okay. And the surrogate mother, acts as this thing and nourishes the embryo, and then brings it to term, okay. Now how does this happen? We are going to see a short video maybe later in this thing, as a short video, okay. Typically you would hold the egg, okay, unfertilized egg, with a vacuum suction device, okay. These are extremely minute, so this is visible under a microscope, so you can imagine how tiny these must be, okay. And then you have a needle which is all of these are extremely sophisticated techniques, okay. This needle again is a very fine needle, okay. And so you can actually get into the egg, you can puncture the egg, okay, and you can remove the egg, and then you can come back with the nucleus from another one, and you can inject the egg back into the enucleated egg, okay. So in this case you can actually see, this is the suction device, this is these are the nucleus, okay, which has been isolated from some other organism into which you want to clone. And this, after sucking it up through the needle, you are trying to inject it back into the nucleus. So this is 2n, n plus n, 2n, okay. And once you inject back into the enucleated egg, the egg starts to expand, okay. So it is laborious, the success rate is extremely low, okay. And these enucleated eggs are fused with, sometimes they are fused with other cells, okay. And the most famous example of this was Dolly the sheep. which was the first mammalian clone from an adult donor cell. Okay, adult donor cell, as we all know, has 2n number of chromosomes, right? Okay. Now, in addition, several animals, were eventually cloned. Monkeys were cloned. Of course, the most popular organism is lab mice. So lab mice were cloned. Pigs were cloned. Okay, many different. The list is now big. Actually, if you go to Wikipedia on animal cloning, you will see a huge list of animals that have been cloned in subsequent years. That means the technology or the technique by which these can be done has become so widely standardized that it is now uniform. It's more or less uniform, and these procedures are to a large extent there fails. You may fail at many steps, but by and large, in well established laboratories, you have standard operating procedures, so you are kind of saved from failures to a large extent. As long as you follow the SOPs, the success to a large extent is assured. Okay, so cloning has become extremely what should I say, common technique now. Just as when keep giving you the example of PCR. When PCR first came into the scientific world, very few laboratories in the world could do that. Okay, then people realized the drawbacks, and people started working on it. Tag polymerase came into the scene, and then the technology for making oligos matured. Eventually, now we can do PCR literally in schools. Now, not even colleges, not even universities, but PCR can be demonstrated in schools also. Okay, and so this is another example. Just as so what is surprising, or what was this thing was that although laboratory mice are well studied, somebody dared to do this in sheep, and they produced a clone. So that was the biggest thing. Okay, and so this is showing schematically what happens in the mouse. Okay, so you basically take the egg and its surrounding cells from the ovary, and the cells are separated. Okay, the nucleus is removed through microsurgery from the egg, and then the nucleus from the donor egg, from the donor, is injected into the egg. The egg is implanted to the surrogate mother, and 21 days later you have a cloned mouse. That means it takes less than a month to clone a mouse. Okay, less than a month to clone a mouse. Of course, if everything goes well, it's fine. Otherwise, you have lots of places where things can go wrong. Okay, but there are still, although we call them as clones, there are lots of unresolved issues. Okay, for example, clones don't live as long. We don't understand why clones don't live long, but we now have some clue why clones do not live as long as the parents. Okay, second, it does seem that although we call them as clones, they are not identical carbon copies. They are not identical. So there are differences. Although overall, overtly, it looks like a clone, they may not be a clone. Okay, some of these clones develop diseases, unusual diseases early. For example, Dolly the sheep, was the nucleus was taken from a donor which had already, passed 6 years. And the donor was supposed to live for 12 years. So right in the middle of this thing, they took the nucleus, and Dolly the sheep lived for another 6 years. Ideally, it should have lived for 12 years, but it lived only for 6 years, and it died of a disease which at that point in time people thought was rare. Okay, but now it is not considered rare. People are still, a lot of questions about Dolly's death. Okay, very low success rate. It's only like 0.1 to 0.3%. That means you have to do hundreds and hundreds of micro injection in order to see a good this thing statistically successful rate. Of course, not many people are so well trained to do this. That means you already have a big challenge. Okay, And it does look like when you take the adult nuclei and transfer it into a egg, you may have incomplete reprogramming. Okay, as I said, once the 2N nuclei is injected, the egg behaves as though it got fertilized, right? And it starts to reprogram itself. Sometimes this reprogramming may not be complete or may not be accurate, and so that is one reason why the clones may not be identical. And it is also possible that this could be one of the reasons why the clones may be suffering from some kind of a disease. Okay, so we will look at a short video. So this cloning has a lot of implications, right? It has a lot of implications. So let me throw a question at the class. Can we clone humans? I'm going to give the this thing to class. Can we clone humans? Devanshi, we will start with you first. Am I audible? Hello?
Yes, sir, you're audible.
Okay. So can we clone humans? Yuvraj said yes. Okay. Anybody else?
So I also think it's mostly possible, but like you said, the success rate would be quite low, and we'd also have to maybe find a way to immunize the clone so that it doesn't easily die of a disease. And so there are many factors to consider, which would explain why, maybe it hasn't been successful yet, but that doesn't mean that it's not entirely, impossible or possible.
Okay, good, good, good. So there are a lot of ethical issues. Tomorrow we will talk about it. Okay, tomorrow's class. There are lots of ethical issues in human cloning. Okay, lots of ethical issues. In fact, once the this thing cloning became Dolly became a reality. Literally every government, the US government took the lead, followed by the European Union, and then several other governments took a lead in banning human cloning. Okay. In spite of banning human cloning, one researcher in China said he has cloned. Okay, and then that became an issue actually. Okay, it didn't come out in the this thing, but there are reports not only in China but also from European Union that they have cloned humans. Okay, but as I said, legally you cannot, and in fact those people who said that they have cloned have gone. No more news is available. Either it's a fake or they are facing some consequence. I don't know which one is right, but you don't get to hear from them anymore. Okay, but there are lots of ethical issues in cloning, but cloning can be very very useful, as I said, when you have endangered species. Okay, and the video that we are going to see shortly talks about that one only. Okay, we will talk about Devanshi, I got your response. Okay. Mm, let's see. Yeah. So, so this talks about the on Jan 8, 2001, a healthy baby gore, a large ox-like animal whose populations are now threatened throughout much of their native range from India to Southeast Asia, was born in Iowa. Okay, and this was born to a surrogate mother named Bessie, and the baby bull, which was named Noah, was the first clone. Okay, of an animal. Okay, so we'll just see that short video. Okay, and this is basically, as I said, involves micro injection, These are ways by which we can introduce foreign DNA, which you remember when we talked about some of those techniques, I stopped short of these things. After transformation, we stopped. You know, these are other ways by which you could transfer nucleic acid. And this is one of the ways which we will talk later. Okay. So, is the video visible?
Yes, sir.
Audio is also audible, right?
Yes.
Okay. Okay. Looking at some of this is kind of graphic. So if you don't want to, then you could, either pause it or fast forward it. Some of it is really tough to see.
We'd planned the day that we would want this very special calf to be born. All our plans were aimed at getting the fetus mature enough to live when it came out in the air, and yet not so mature that it would come out by itself and we would miss it.
It's been a long 10 months. I lost a lot of sleep thinking, what happens if this animal doesn't make it? This could be a severe blow to the potential use of cloning technology in conservation, and felt like the whole world was kind of watching what we were doing. So I was pretty nervous.
I'm not getting it out this way.
When I first heard about this plan to clone a gaur, I was very intrigued. A gaur is an ox-like animal that lives in Asia, and it's actually threatened in the world. There are very few individuals left. Somewhere between 13 and 30,000 are left in the wild. I think the fact that this species is a threatened species added a different element to the whole question of cloning. It's very different if you're using it for conservation than if you're using it for domestic species.
It's going to be a tough one to roll over, Jose.
Jeez. We got Noah's genetic material through a third party called Rescue. His cell line originated from the San Diego Zoo. The gaur died in captivity, I believe, in 1992. So the tissue sample was cryopreserved or frozen for about 8 years. What we wanted to do at the laboratory in Worcester was to apply a principle of somatic cell cloning, but to kind of
So
put
you
a
can see
slight
everything
twist on
is
it by
done under
doing a procedure
microscope.
what we call
Okay.
cross-species nuclear transfer or cross-species cloning. And what that involves is taking genetic material from one species and fusing it to the egg of another species. We had a whole group of mature cow eggs that actually have cow DNA inside them. For us to be able to utilize those eggs, we have to remove the cow DNA. We then picked up an individual gaur cell that contained a nucleus that had gaur DNA present. Placed that cell into the egg. Applied an electrical pulse which allows the two components to fuse together. We then apply some chemicals that induce what we call fertilization events, and that embryo then starts to divide. We keep those embryos in culture for 7 days.
मैं class में हूँ.
We then ship the embryos back to Iowa at Transova Genetics, which they then transfer these gaur embryos into a domestic cow.
Got it.
So the whole project was done without ever having to come in contact with the living gaur. And actually that's important when you're looking at this in the field of conservation. There are so few of these animals left. Many zoos don't want us doing research on their gaur population, so we were able to actually produce a gaur without ever having to touch a living gaur.
That amnion's just a bit thick. Let's get him. You got the cord?
Come on here. I got the core. Okay. Cool. Yep. Cool. It's coming. Cool. Come here.
The delivery itself is nerve-wracking for two reasons. Firstly, it's always a little bit tense because you don't know what is going to come out of the recipient animal, how healthy it will be and how much work you have to put into resuscitating it. So it's always a tense time when you get a neonate, a newborn animal, and you have to see whether it's going to breathe. But it was even more special in this case because it was such a unique animal, and it's the only one that we had to work with. If this one didn't live, there was no second chance. It would take another year or so to get more pregnancies on the way.
I think this type of procedure can be of some use in conservation biology. As a scientist, I'm aware that habitat preservation is extremely important and biodiversity is important for survival of a species. What we were trying to show with this technology was that potentially this could salvage genetics that would have been lost in a population. So in Noah's case, he was actually derived from a gaur that had not reproduced or allowed his genetics to go on to the next generation. We're now able to technically salvage that genetics that would have been lost from that male gaur.
Cloning has the possibility to bolster diminishing populations of animals. The question is, should it be used? My worry is that the public and politicians may think of this as an easy fix. Why conserve animals in the wild if we can go ahead and clone them in the future and replace them? The problem is that's absolutely not true. Biodiversity is so complex, and the interactions between species and their environment and within species are things that we can't replicate, as smart as we are and as much as we try. And I think the safest and best thing to do is to let biodiversity alone and continue as much as possible, rather than thinking that if we put our energy and effort into something like cloning, we can somehow replace what we've lost.
Bessie doesn't have a calf to look after, but she won't be missing that too much. She's not bellowing. She's recovered from the surgery, recovered from the C-section very well. And Noah is looking great because he can stand up. He's very alert and active. He's not as advanced as a normal calf would be at 12 hours, but we think he's doing just fine.
I'm kind of tired. It's been a long couple of days, every couple hours coming back, checking on the cow and then checking on the calf. And I think I'm still slightly in shock. Every time I go and check on Noah, I look at it and my first impressions are, oh, it's another cloned calf. And then I stop to think, I'm like, well, wait, no, this is actually a wild animal that's sitting out here. And then I think about it, I'm like, yeah, it's the first cloned endangered species. And it feels pretty good.
Okay, so tragedy, right? 2 days after the birth, the baby died of bacterial infection. How it survives in the wild but not in the lab, where things are controlled, is still a mystery, right? In spite of giving the best care, in spite of giving the best possible environment, the baby died, and that is why one reason why the success rate is low, right? So not only for plants, you could do this for not only for animals, you could do this for plants also. species which are endangered, right, can be cloned by tissue culture. Of course, I think plant tissue culture is a totally different field by itself, and there have been a lot of advancements in plant tissue culture, and so anything that is possible, I'm sure you will get to hear from your other faculties. But when it comes to animals, I think cloning has been a boon, right? People are actually trying to resurrect the woolly mammoth, which was found frozen in the Siberian, ice caves, right? And people have been trying to see if they can clone few other animals. As of now, nothing has been successful, and so the chances of a Jurassic Park happening in our times is probably remote. Nevertheless, anything that is preserved well, has the possibility of, getting cloned. Only thing is, at this point in time, although the in vitro fertilization technique and every other technique is okay, we still don't understand what should be the characteristic of the, the egg and the recipient eggs. Okay, so we have to. Hello. So it is. I'm in a class. Can I call you later? So it is going to take this thing, some more effort for us to understand what should be the characteristic of the donor egg and all that. Okay, but irrespective of all that, as long as you have the species to which you are transferring the egg, as long as it is closely related, it does seem to work very well. Okay, so we will continue our. So, any questions on today's class?
No, sir. Yes, sir.
Go ahead. Somebody had a question?
Oh, yes, sir. Am I audible?
You're audible.
Sir, so we already had a process known as embryo splitting. So, what was the need for a more complex process like nuclear transfer? Does it have more advantages?
See, embryo splitting is only when you have, for animals that are already there, right? Things like gore, which are like from wild, right? Which the numbers are diminishing. It's very difficult to, observe their this thing, right? You need to collect, harvest eggs. You need to, get the embryos formed, which is a fairly difficult process. So this single cell nuclear transfer is more useful when you have endangered species, right? Otherwise, this was more like when Dolly the sheep became the this thing prototype for cloning. It was just showing you the power of this technology, right? And since then, as you rightly pointed out, if embryo splitting is an easy way, people would resort to that. But since for endangered species, it is very difficult, right? Going into forest and chasing these wild animals which are endangered is a very difficult process. So ideally, if you're able to catch single endangered species, take the somatic cell, take the skin cell, for example, take the nucleus, put it into a related organism's egg, it is going to grow. So it becomes easier that way. That's the only reason why you have this animal cloning.
Okay, sir. So, one more question.
Yeah.
So, sir, if a plant or an animal is suffering from a disease, will its clone have the same disease?
It is possible. It is possible, as long if it is an age-related disease, for example, uh, you know, um, it will probably be fine in its normal, this thing. And once it becomes aged, it might get the disease. Okay. On the other hand, if it's a genetic disease, for example, then there is nothing at this point in time we could do. We can only do, of course, there are a lot of advancements in molecular biology. You all must have heard about CRISPR-Cas9, for which this year's Nobel Prize was given. So we could do corrections with CRISPR-Cas9, but getting an embryo itself has very low chance. Then getting CRISPR-Cas9 to work on those is going to be even lower chances, right? So people are still trying to refine the technique, but it is possible that at some point in time that you could, take an endangered species which is diseased. You could correct the disease and you could, get it back on this thing, normal this thing. It is possible.
Okay, thank you, sir.
Good question, Shakti. Any other question? Okay, so most of these, so what we saw today was reproductive cloning, right? We will talk about therapeutic cloning in tomorrow's class, okay, which essentially involves, taking the patient's own cell, okay, reprogramming it to become more like embryonic this thing. And then once so an embryonic cell, the biggest this thing about an embryonic cell is that it can become any cell in the body, right? You could take your skin cell, make it like an embryonic cell, embryonic stem cell, and then after you grow them for a while, depending on what kind of factors you give, it can either become a liver cell, it could become a lung cell, it could become neuronal cell, it could become any other cell. Okay. How do you differentiate them? How do you differentiate them from a normal cell into these nerve cell or hepatocytes or anything? It is based on the growth factors, right? For example, almost all the nerve cells have nerve growth factor. You will never see nerve growth factor on your skin or on your stomach or any of those things. Those cells do not express nerve growth factor, but neurons express a lot of nerve growth factor. So if you were to include nerve growth factor in your growth medium, there is a high chance that those cells will become nervous cells. Okay. And if you include what is called as hepatocyte growth factor, then most of the lineages will become hepatocytes or liver cells, right? So depending on each cell has its own marker, growth factor. Epidermal, for example, you want to make a cell into an epidermis, or which is useful for burn victims, you need to add epidermal growth factor. Then your cells will become, most of them will become this thing, okay, useful for skin, keratinocytes. They all will become keratinocytes, which can use it for your therapeutic cloning. Okay. So we'll look at all those things in tomorrow's class. Okay. Any other question? Okay. If there are no questions, that's fine. Any questions, I'm willing to answer. All right. So we'll meet tomorrow then.
Thank you, sir.
Okay.
Thank you, sir.
Yeah. Take care. Bye.